Hi, I'm Brian Douglas from Bento Lab. In this video, I'm going to be showing you how to extract DNA for PCR using the hotshot method. The hotshot method is a simple two-step protocol performed in a single PCR tube. It is quick, cheap, and it can work with a range of different samples. I'll be using the Bento Hotshot DNA extraction kit. The kit comes as two 30 milliliter bottles. One is the alkaline lysis solution, and one is a neutralizing buffer. Together, they're suitable for 400 DNA extractions if you use 75 microliter volumes. Let's get started. First, prepare your workspace. Before beginning any experiment, it's really important to clean your workspace and tools. This will help you avoid contaminating your samples, your reagents, and help you get better results. For sampling, you could use tweezers and scalpels. You can also use a double-edged razor blade as a disposable item. You can snap the blade in half in the packet and then use a single half per sample. Before you start, make sure that all of your tools in your workspace are clean and fairly sterile. This will help you avoid contaminating your samples, your reagents, and help you get better results. Have a look at our guide to clean sampling practices to find out more. Next, document your samples. Here I have a piece of lamb liver. Label your sample tubes with a permanent marker on the tube lid and on the side of the tube. I'm using PCR tubes, which are 0.2 milliliter tubes, so that I can do the incubation step in a thermocycler. The first step of the hotshot method is an incubation step in hot dilute sodium hydroxide. This involves heating the sample in a 25 millimolar solution of sodium hydroxide, which will rupture the cell membranes and release DNA into solution. To do this, add 75 microliters of alkaline lysis solution to each tube. If you have multiple samples, the same pipette tip can be used for all of the tubes. If you have very small samples, you can use a smaller volume of buffer, for example 50 microliters. Now we're going to add the samples. Using the sterilized tools, remove 1 cubic millimeter of tissue from a sample. It's really important not to add excess material because adding too much sample is likely to cause the extraction or the PCR to fail. 1 cubic millimeter is about the size of the end of a mechanical pencil. Transfer the sample into the labeled tube. You can gently flick the sample into the tube or place the sample directly at the bottom of the tube using tweezers. Make sure the sample is immersed in the alkaline lysis solution. If the sample is not immersed, you can try flicking the tube again or briefly centrifuging it. For very stubborn samples, you can use a fresh pipette tip to move it into the bottom of the liquid. If you're using the hotshot method for multiple samples, remember to sterilize your tools in between each sample. Now for the hot step of the process, the heat incubation. I'm using the Bento Lab thermocycler as a heat block, but you can use other thermocyclers or heat blocks with a heated lid. Check that the tube lids are securely closed, then place your samples inside the thermocycler. Incubate the samples at 95 degrees for 30 minutes. Once the incubation step is finished, check that all of the tubes remain tightly sealed. Allow the samples to cool to room temperature. You can also cool the samples to 4 degrees using an ice block or in a refrigerator. Now for the neutralizing step. We're going to be adding the neutralizing buffer to the solution to reduce the pH from 12 to around 8, so it can be used in PCR. The buffer also helps to stabilize the DNA so it can be stored for later use. First, add 75 microliters of neutralizing buffer to each tube. If you used a smaller volume of alkaline lysis solution earlier, then you should also use an equal volume of neutralizing buffer in this step. Mix the solution by inverting the tube several times and then flicking the tube with a finger until it's fully mixed. If samples are murky with particulates, you can briefly centrifuge them to sediment the particles to the bottom. This final solution is a DNA extract. You can use 1 to 5 microliters directly for PCR or store it until needed. The solution has a pH of around 8 and it contains 50 millimolar tris hydrochloride and 0.25 millimolar EDTA. This is similar to standard DNA storage solutions, so you can refrigerate it for short-term use or freeze at -20 degrees for longer-term storage. Once you are done, make sure to put the hot shot bottles back into the bag and seal it. To maximise the shelf life of the solutions, it's important to keep them away from carbon dioxide, as this will slowly neutralise the lysis solution. The bag adds an extra layer of protection against carbon dioxide in the air. You can find a written version of this protocol on our website. And if you'd like to read the original paper, or find the Bento Hotshot DNA extraction kit, you can find all of the links in the description below. If you found this video useful, please give us a thumbs up and subscribe to our channel. If you have any questions, or if you are using the hotshot method yourself, please leave a comment. We would love to hear from you. Thank you.